So first of all, very good morning, everyone. So now science and technology and environment. I will also take environment and ecology, and I am going to target a coming prelims exam 2024. So what I have done here is that few of the topics I have covered last time also there. When I came the last time, I took on science and technology, but not in environment and ecology. But this time I will be targeting environment and ecology topic also, which are relevant for the examination. And you all know that every year around 15 to 18 questions are asked on ecology and environment, right? So that part I will be covering in the classroom. Plus science and technology, again, around based on current affairs and all the latest development in science and technology. Again, you get questions something around 12 to 15 questions. So we'll see all the dynamics part of it. So what I'll do here, one by one, I will be going to certain areas. I have kept certain topics here just for my reference. This is from the magazine, from Amigos IAS magazine. I have just asked them to just filter out a few topics from there so that I can cover and discuss in the classroom. But let me go into certain topics here. I will get back to the things what is given here. But let us go to the main part of it. See, one area where you get questions in the coming exam, right? We will be getting the question regularly last 5-6 years. There is a biotechnology part. So let me discuss certain areas, then I'll get to the main part of it. See, when it comes to biotechnology, last time I have covered few areas, but let me discuss certain more topics in biotechnology for the coming exam. What questions you can get there. See, one part which is making a lot of headline, that is a genome. That is a DNA sequencing. When you look at genome here, there is a total DNA content of a living organism. That is what called genome, gene plus chromosome. Like you know that every living organism with a cell has got a genetic material in the chromosome, and chromosome is made of a nucleic acid DNA, and within the DNA there is genetic information in the form of genes. So when you come to genome here, the first thing which comes here, whole genome sequencing. So there are a number of projects which are going around worldwide to sequence the entire chromosomes of a living organism. Let's say human genome project, HGP, or let's say, for example, another organism, right? So whichever living world we talk about, when a scientist do the DNA sequencing, the one thing is that the whole genome. What is whole genome? Total chromosomes, total DNA. They are sequenced in the lab to understand the molecular basis of life. Now, from the current affairs, what is very important, which is making here, the first thing is this one. First time in India, jamun, jamun tree, right? Jamun, nowadays in summer you have mangoes, like mango and all is it. Jamun is another fruit which people eat in India. The jamun has been now sequenced, right? Let me come to that one. So one thing which has made headline, that is a jamun tree. This DNA sequencing has been done. Now, why scientists selected this particular tree? The reason is that jamun has a certain medicinal properties. Like first is the anti-diabetic. Like diabetes, diabetes is caused because of the lack of insulin and the high level of blood sugar. So jamun as a fruit, if a person suffering with diabetes eat the jamun, it can lower the blood sugar level. Jamun also has another property, antioxidation. In the food what we eat, our body produces the radicals, free radicals. They're neutralized by so let me write here. So if you look at jamun here, one property is this one, anti-diabetic. It has got anti-diabetic properties. Another one is antioxidant. Like in biology, like science, technology is connected with biology part also. Biotechnology is connected with biology. So when you read biology, you will come across vitamins and minerals. And one vitamin is called vitamin E. Vitamin E has got antioxidation property, right? Tocopherol, that is a scientific name is tocopherol. So Jamun has got, but what are we talking about? We are talking about DNA sequencing. So why scientists selected Jamun? Because Jamun has got certain medicinal properties, and scientists would like to understand those properties based on the genetic aspect. What is something called gene, or what we call a marker, biomarker, and that can help us develop certain more new varieties of medicines in coming time. Future medicinal development could be there. So one is that whole genome, we come across this one, Jamun tree, which made headlines. Now another thing which I would like to add here, see, when you say human genome, so there are a lot of projects going on worldwide in terms of one is that which was originally done in the US, but now there are group genome projects are being done. Like for example, there is a one thing which India is doing, IndiGene. See, one is that first time entire human genome was sequenced in USA, right? That was in 2001. That time the entire blueprint was brought. But now I'm bringing some current aspect there. Based on the same kind of DNA sequencing, we are now doing the profiling of humans all over the world. And one of the very landmark project is IndiGene. That is in terms of doing the genome sequencing of Indians all over the country. So this is actually a group project. It's not a individual project. The IndiGene, that is a here mapping genomes of Indians. Now this has been in the news for a long time. I'll add some international project also. So one of the very leading project which is happening in biotechnology field, genome sequencing, that is IndiGene, where you know that we have a very important organization in Hyderabad, CCMB, Center for Cellular and Molecular Biology. Now the team of scientists are doing mapping of genomes of Indians all over the country. And you know that we have a racial, if you look at race, ethnicity, we have diversity. If you look at from northeastern side, then northern side, southern side, we have a huge diversity among Indians found there, what is called variants, right? Different type of variants. Now, why the scientists want to understand this kind of a molecular basis? One thing, you know, what is the application here? Understanding the disease, prevalence of disease, right? Even it can help in terms of genetic diversity in terms of human race. Like, you know, for example, anthropology, okay, how the migration pattern, how they different, like for example, Andaman and Nicobar, you know, Jarawa, Shompen, there's so many tribes are there. How they are linked with the tribes in Africa, or let's say Europe, or let's say Americas. Again, we can understand through molecular basis. So that type of research that is being done. Now another thing is this one. There's another project called 100K genome project in UK. What is the name of the project? 100K. K means 1000. So now UK, United Kingdom, they are carrying out a complete genome sequencing of the citizens of UK. Again, you see the UK, although you have read in Indian history that Britishers, colonial rule, they were ruling the many parts of the world. Even India was also under the colonial rule. And you know that after end of colonialism, UK was very liberal to accept migrants. And today, if you go to UK, now it has got a mixed type of population. You know, even the Indians, even the Prime Minister, you know, PM is also from Indian origin, right? You are aware of the name there, right? So if you look at, there are so many people who migrated there and lived there. And it's not only from India, Asia, you'll find Africa. So now they are trying to understand genetic profile of the UK citizens. There is a 100K project is there. Then there is another project which is very, very important from human genome understanding. That is a 1000 genome project. This is being done profiling at the global level. 1000K, that is done being global level. So all these projects are making headlines. So now here what I've used the word whole genome. That means complete set of gene, complete set of DNA. So somebody's mic is open there. So what I was saying that when you look at such kind of projects, they are trying to understand the entire aspect of disease, the variations that's coming there. Now the thing is that when you read current affairs, you should always think of the questions. Okay, what kind of questions can be asked in the exam? How UPSC is going to ask the question? There are two levels of question. One is the direct name of the project, and you will ask the question ABCD. Second, what the question can be asked in the statement type, like what are the applications part of it, right? Where this kind of a project can play a very significant role, that could be part of. So there's one area which I would just like to add here. Now, when you look at the part of another thing here, see this I've given about whole genome, right? Number one, which I've done there. So now let's look at here. Another thing which is making a lot of headline. Sometimes the scientist may not be interested to learn about the entire chromosome, entire DNA. Sometimes the scientist would like to just understand certain part of the organism's DNA in terms of identifying any kind of a property. So now what is here coming here? Cell-free DNA. I think this I covered last time, right? Just I'm again repeating this, cell-free DNA. Now what is cell-free DNA? Let me bring about the main aspect. One is that, let's say these are DNAs obtained from body fluid. Let's say, for example, such as blood, or sometimes, brain. If you look at, we have a very important fluid called cerebrospinal fluid, CSF. The such kind of fluid or liquid part of the human body, which may have some kind of a DNA, and those DNA, this DNA from either dead cells, or sometimes what happen, fragmented cells, or ruptured cells. Of the body. Sometimes what happen, our body may suffer internally, internal damage. There may be some kind of internal damage. Sometimes, blood clot. Sometimes the people may have a hemorrhage, fever, hemorrhage, some kind of internal rupture. So there what will happen? That entire body organ is lined up with cells. Let's say lungs, suppose lungs. Lung is made of cells, kidney made of cells, liver made of cells. Suppose liver damage. So liver has a liver cell. Now when the cell will die, within the cell there is a nucleus. A nucleus contains chromosome and DNA. So those DNA will be in the body fluid. So that, let's say brain, suppose brain hemorrhage, brain damage. So whatever the damaged cells are there, its DNA will be the part of the CSF, cerebrospinal fluid. So through those DNA, doctors or scientists will be able to identify the cause of disease. So what is very important here? This basically helps in diagnosis. Of diseases. Let's say, such as one area is this one, cancer. One area which has become very prominent in this context is cancer. Tumor and cancer. Let me minimize it. People who cannot see, I'll just take it upward. Now it is visible to everyone. Let's say one could be the tumor and cancer, right? Or let's say, for example, this one, organ damage, let's say kidney damage. Or let's say sometime blood clot. So scientists will be able to understand those causes of the diseases, the problem in the human body. Now this is another area of what is something called molecular diagnosis, where we are using biotechnology. And today biotechnology has advanced to a stage where in the lab we can take the sample and we can have DNA analysis. What we can have? DNA analysis. And we can identify the case of that. So you know that if you look at previous year questions, PYQs, UPSC asks question biotechnology from two perspectives. One is the current aspect, second is something which has not been previously asked. That means UPSC is not going to repeat the question. See, genome is making headlines, and something which is very obvious. You know that the exam has become so difficult, right? With the kind of a result which has come up with the cutoff. Now you know the cutoff also there, the kind of question which is being asked in the exam. So that's what I said. When you are reading current affairs, you should know what is there in current affairs. But go back to the concept. Get back to the concept. Okay, how the UPSC can ask question. Any kind of question can be asked. That's what I say, that if you understand the things, it will not be difficult for you to solve the question here. Right? So this is one area which is another thing which is making headlines for the last so many you know months, and it could be a question in the exam. Cell-free DNA, right? One is a whole DNA, entire DNA you come across. Now another thing which I would like to add here, I have mentioned last time, but again, since there may be students who are new to the batch, mitochondrial DNA, mtDNA, right? DNA from mitochondria, because mitochondria is a cell organelle. Its primary function is a respiration, aerobic respiration, but even it contains a DNA which can really help understand certain basis of human being or other living organisms. So so I'll change the page here. So I've just mentioned about this one, self-free. Second one, I'll do one thing here. Let me take to the third point here, mtDNA, mitochondrial DNA. See, when we talk about whole DNA, whole genome, we are taking into account not only chromosome but also mitochondria. See, when the scientists do the DNA sequencing of a living organism, so it will do the sequencing of entire chromosome, all chromosomes, including mitochondria, right? But sometimes scientists can do DNA sequencing only of mitochondria, not the entire thing, only mitochondria. Now here, there's something called this one, cytochrome C. I'll explain what is cytochrome C. Within mitochondria, let me draw a diagram here. When we see under microscope, electron microscope, mitochondria looks like this. This is the structure of mitochondria, right? What I said, what is main function? Respiration, breakdown of glucose to give us the energy, ATP, what is called adenosine triphosphate. Now within this, you have a certain type of complex called cytochrome, and this is involved with the respiration process. Cytochrome is a part of the mitochondria which play a significant role in terms of having the kind of a respiration process. Now this DNA, or you can say the part of the information, gene. What is gene? Sequence of DNA which has certain information about any aspect of that particular organism. Now here, if you look at this one, there's something around 37 genes only there. See, if you take human being, mitochondria, we have whole DNA. If you take whole genome, we have around 30,000 genes. If we take man and we take all chromosomes, we have 30,000 genes. But if we talk about mitochondria, 37. So if we compare, ratio wise is very small and doesn't have so much of does not have any significant aspect on traits of human being. I'm talking from the human being point of view. Human beings. You all know that this all mitochondria we get from mother. How we get from mother? Because of egg or ovum which get fertilized, right? From there the mitochondria is obtained. Now these 37 genes don't have so much of significant role to traits. What are traits? Our characters. But if these mitochondria are defective, then it causes disease in human beings, right? So if they are defective, these genes cause disease. What I want to say, significantly, if you look at our phenotype, what is phenotype? What we are made up of. Physical part of it. It doesn't have so much of role. But if something goes wrong with this kind of genes, then the person may suffer with some kind of a diseases. Sometimes, autosomal, or sometimes immune diseases, autoimmune diseases could be possible. Sometimes the other kind of genetic disorder could be possible. So you already know that which UPSC has asked question. See, again, you should know the previous year question, but you should always be knowing that where question can come, where the future question can be asked. But just I'm bringing here, UPSC has once asked a question about three parents baby, right? Mitochondrial replacement therapy. So one area, like for example, there's a disease called Lie disease. LEIGH, Lie disease. This happens in humans. What happens with Lie disease? The child who is born is not able to survive for long. The person dies after the birth. So first time you are aware, the UK, United Kingdom, carried out mitochondrial replacement therapy. That is what called three parents baby. So that was the replacement part. So here, so I'll write here, you are aware of this particular one, but already question been asked. Remember that this is already question has been already asked in the UPSC exam. I don't think so UPSC is going to repeat the question. This just I'm giving the information which you are aware of it. Replacement therapy. So let me interact with people here. What basically they did in the lab? First time where it happened? UK. UK was the first country. Anybody in the classroom? Then I'll come to other aspect of mitochondria. Mitochondrial DNA, one thing was, this was asked in the exam. So what exactly the scientists did in the lab? What they did in the UK? Huh? Huh? Yes, what he has said, I'll just highlight that. Mother's mitochondria was defective. So what they did was, they replaced the defective mitochondria of mother with a donor woman, another woman who donated the right mitochondria. That was replaced. And there's something called technique called pro-nuclear transfer. They use a technique called, let me write here, this technique is called pro-nuclear transfer. Already this question has been asked in the exam, but just I am bringing here into the classroom here. They use this pro-nuclear transfer technique. They replace that mitochondria. But again, remember that this is not the only thing which was done in UK. After UK, the other countries also followed it, right? But it was allowed under the law. That means the government went to allow this kind of changes. Under ethical guidelines, this was allowed in human beings. So that's what I brought here. But now let's understand other aspect of it here. Mitochondrial DNA has very important role to play in environmental aspect, what is called here DNA barcoding. DNA barcoding, I'll write on the board, DNA barcode. And what has made headlines in India? We have now DNA barcode of tigers. India is the home of largest population of tigers in the world. We will cover in the environment topic. We have a highest number of tigers. That means more than 70% of tigers worldwide is found in India. So we have done a massive exercise of DNA barcoding of tigers. Before a few years, Bangalore, Bangalore has got one national park. Can anyone tell me which is a national park in the Bangalore city? It's very near to that. Like we also have in Hyderabad national parks. Can anyone name one very important national park in Bangalore? Bangalore is in Karnataka. Name any national park which you are aware, which is in Bangalore. But now, it was earlier outskirt of Bangalore. But now, Bangalore has expanded like anything. Like same way, in Hyderabad, we have a Mahavir, Hiran, this one. But now, the Hyderabad has expanded so large. So now we have come to the very vicinity of that national park. Same with that Bangalore National Park, the Bangalore has expanded. It was in the news. Can anyone give me the name of the, I'm asking GK question, general knowledge. Banerghata is a very popular this Bannerghatta National Park, that is in Bangalore. Now what I want to bring here, long way back, is almost around 10 years back. Bangalore, this Bannerghatta National Park is known for butterflies. Butterfly park. In India, we have butterfly parks. We will cover in the environment topic. And even that could be a question in the coming exam, butterfly park. Nowadays, certain type of butterflies are now threatened, critically endangered, ready to book. So Bannerghatta National Park, so there butterflies which are found here. This has been done. DNA barcoding. I'll be coming to that. Just give you the example here. Now, why Bannerghatta was in the news? Let me connect with the environment. It was in the news because there's one provision you are aware, ESZ. What is ESZ? Eco sensitive zone. That was declared by the government around Bannerghatta. Why government take that kind of action of ESZ? Because of development outside the park to control the human activities. And since I've told you that this was in outskirts of Bangalore, but now the urbanization in Bangalore, the way of human population growth has come very near to the park. So recently government declared ESZ around Bannerghatta National Park, and there was a protest with that. Protest because if you have a land, suppose you have a land or you are living around that area, and now government put some kind of restriction. So definitely the people will be not so happy with the development. So that was in the news. But here I am giving the example from biotechnology, where the mitochondrial DNA of all such butterflies have been mapped. So we have now profiled something called profile of butterflies. So this is the first very good example of project which the government of India did for butterflies. So now recently another thing is this one. India has created tiger DNA barcode. This is what we have created. So it means what? Whether it's a tiger or leopard or you take elephant or any threatened species, we can carry out DNA barcoding. And now once it was asked in the exam also there, the applications of DNA barcode. So what I'm now coming here, when you talk about DNA barcoding, is basically again you have this one identification of a species based on, I write in short form here, mitochondrial DNA. Based on mitochondrial DNA, any living organism can be identified. Now it has a very important role to play. I'll write here, if you take the application part of it, MT is a mitochondria. So if you take the use of this kind of a method, so the uses or applications are, one is this one, census exercise, like tiger census, elephant census, lion census. So we can identify one tiger with another tiger, because mitochondrial DNA may give us idea. And sometimes fecal material, sometimes dung, like dung or fecal material. From there the scientists can take the DNA and in the lab can find out whether it's the same tiger or different tiger. Again, sense exercise. Second thing, what comes? Second comes this in terms of illegal trade. You know that from the wildlife angle, biodiversity and wildlife angle, India is again known for illegal hunting, poaching, and different parts of the animal, they're illegally sold in the outside market. So then again, custom department, tortoise, turtles, then sometimes butterfly, even butterfly, exotic type of butterflies are illegally traded. So one can identify the sample, and you come across the newspaper sometime raid the Wildlife Crime Control Bureau, they raid the premises, they capture the such kind of samples. So again, the mitochondrial DNA and the DNA barcoding can play a very significant role controlling such kind of trade. Okay? You know about red sanders, you're aware of red sanders. Even that kind of DNA barcode can help. So illegal trade control could be there, which can be found. Another thing where such kind of thing can be used in DNA barcode, that is in terms of a study of biodiversity of a particular area. Normal study, that here study of biodiversity of a national park, sanctuary, that we can do it. Census is counting. You are doing the counting of a particular animal. Let's say tiger. How many tigers are there? But sometimes you want to do the profiling of a particular protected area. Let's say sanctuary, national park. So again, we can use DNA barcoding. That could be giving us idea at the molecular level. Again, this is based on mitochondrial DNA, right? Mitochondria plays a very significant role in this kind of understanding. So what I've done here in the first part of the class, in biotechnology part, I have discussed the whole genome sequencing, and then I have gone into this particular type of cell-free DNA and mitochondrial DNA. Now let me come to another area, which I have touched upon in previous class, but again I'll highlight here. One area which I think could be a question in the coming exam, that is a gene therapy. Anybody in the classroom last time which was there? I discussed the topic in the last time, and it is making all headlines. It's all making all headlines there. You all are writing this one? Please keep on writing. I will come to that document of Amigos. I will be telling you what exactly is there. But first, I wanted to discuss certain topics in the class, right? Along with your doubts and everything. Now let's look at here. Another thing which I would like to add here, number 2, let me make a flowchart to give you idea. These 2 topics you should properly prepare. One is this one. These are 2 names I have written here, which has made headlines. One is Cas-Jave. Now what is Cas-Jave? Let me explain here. This is the first licensed CRISPR-Cas9 based gene therapy to treat, I'm writing the disease also there, sickle cell anemia. Sickle cell anemia and thalassemia. Let's discuss first. What is Cas-Jave? It is the first time in the world, first time in the world where the government allowed and gave license to the company to carry out gene editing. What is CRISPR-Cas9? Gene editing. To treat 2 type of disorders. What are 2 disorders? Thalassemia and sickle cell anemia. Now let's come to the biology part of it. So first you can write it down, the biology part. What is sickle cell anemia? Okay. So let me write here. If you take sickle cell anemia, genetic disorder, what happens? The normal RBC is disc-shaped. Right? This is a biconcave, concave kind of thing there. We have this kind of a shape of the RBC, red blood cells, which carry the oxygen, carbon dioxide. The person suffering with sickle cell anemia, it converts like this. Shape changes to sickle. The natural RBC, which carry the oxygen, carbon dioxide, is this particular one. This is natural one. This is the normal one. Normal one is there. And this is the sickle cell. Now, this is in biology we call autosomal disorder. Now, what is autosomal disorder? The defective gene, the gene which is defective is in the normal chromosome. Let me explain here. We have 2 types of chromosome, autosomes and sex chromosome. You know, X and Y are called sex chromosome, which determine either male or female, right? But other than the 22 pair, 44 chromosome are autosomes, which we have in the body there. And if the gene is defective there, it can also cause disorders in human beings. So sickle cell anemia again is a problem we link with hemoglobin. The main thing is RBC and hemoglobin. What is RBC made up of? Hemoglobin. So that's what problem comes here. So what is the disorder here? Autosomal. Let me write here. Both this comes into autosomal disorder. Other chromosomes, not the sex chromosome, the normal chromosomes get affected. And the parent, anyone the parent, father or mother, carrying the defective gene, which go to the child, and child suffer with that. Now let me give you data about sickle cell anemia in India. If you look at the proportion wise, India is suffering something around 30,000 sickle cell anemia cases there. These are reported cases of sickle cell anemia in the country. That means rare. What we say? Rare disease. If you look at billions of population of Indians, 30,000 is nothing. And this is more predominant in tribal population. Like, Chhattisgarh, Odisha, Jharkhand, tribes. And you can link again with anthropology. You can link back to the Africa. In Africa, again, the sickle cell anemia is predominant. So tribal population in India, particularly, eastern part of the country. What is eastern part? Odisha, Jharkhand, Chhattisgarh. You'll find predominance of sickle cell anemia. But again, worldwide, this prevalence could be there. Right? Now coming to this one. Thalassemia. And you will come across beta thalassemia. What is what called? Beta thalassemia. Now tell me what happened with thalassemia. If any child is suffering with thalassemia, what will happen there? Hemoglobin. Hemoglobin is not proper. Let me write here. In this case, thalassemia, let me write here. Thalassemia is a disorder, again autosomal. Here there is improper hemoglobin. And the RBC again suffer there. Yes. He's very right. That's called beta thalassemia. See, hemoglobin is made of 4 chain, peptide chain. Alpha and beta. 2 alpha, 2 beta. So there is some defect in the chain of the hemoglobin. Now what happened with this kind of children? They have to continuously go for blood transfusion. They have to continuously go for blood transfusion. Those children who are born to such parents suffering with this kind of disorder has to continuously, lifelong has to go for blood transfusion for survival. Because you know that if hemoglobin is not well, then the person will have problem with the oxygen part and the child will die. So they have got hypoxia. So both the cases have been now treated with this Cas-Jeb. Now let me explain what exactly technique is there. Bone marrow. We have a long bone, like femur and bone marrow. So there we have a stem cell, bone marrow stem cell. So let me explain here. So I'll do one thing here. Just you can write in below here what exactly scientists did here. They edited genes by CRISPR. You know, CRISPR is a very powerful technique there. Like CRISPR-Cas9 of bone marrow stem cell. Bone marrow stem cell. I'll explain how this technique really works here. What is done in this Cas-Jeb? Scientists will take out the blood. See, from where the blood will be taken? Bone marrow. They will take out the blood, bone marrow. Now in the lab, they'll take to a lab the bone marrow blood. Now the bone marrow blood will contain stem cell. That will be modified using CRISPR-Cas9. You know, 2020 Nobel Prize was given to this technique. They'll modify the stem cell to produce normal hemoglobin. Why the child suffer with thalassemia? Because hemoglobin is not properly made. That stem cell has a problem. So they edit the stem cell of bone marrow in the lab. And again the blood will be given back. Now what is good thing here? The best part of this therapy is that it is a one time. If one time it is done, child will be totally free from the thalassemia. Child will be totally free from sickle cell anemia. It's a one time treatment. Here, let me write here. One time treatment, but it is very costly. Remember that. Such kind of thing is very, very costly. It's not simple to have this, and it takes 6 months, 6 to 8 months. and again that edited bone marrow stem cell will be again inserted back to the bone marrow. So again, side effect. Are you able to understand? You are taking from bone marrow. There's a risk of infection, side effect, right? That could be the possibility with the child. So that is a risk. What is a risk? But what is the positive thing? The child will be totally removed from the disorder, cure from the disease, right? So one thing is this one. So when UPSC will ask question, say how the UPSC can ask question. UPSC can give you the names. Suppose the UPSC gave exam, Kazebe, next Kati, some names seen in news are ABCD. Even this could be a simple question, very simple question. The names will be given 3-4 names and will ask what is this ABCD. So immediately in your brain you will know that I have read about this kind of technique. So that means there is a gene therapy, right? Or second, what could be question? It will ask you about gene therapy, right? About gene therapy question will be there. But I'll come to that question also. But now let me go back to this one, another part of it here. Next CAR-T. So anybody, you have read this one. This was in current affairs. All the current affairs magazines throughout the country, they have covered about CAR-T cell therapy. Where it has been done? Mumbai. IIT Mumbai, there they have done for this one. And even Draupadi Murmu, our president, she was in Mumbai in terms of popularizing this method here. And first time Indian patient has been recovered from a type of disorder called leukemia. Leukemia, blood cancer. Now this leukemia is because of genetic disorder. Again, where we use gene therapy in terms of genetic disorder. So let me write here. So what is very important here? Next CAR-T here, this is the gene therapy to treat. Let me write the disease here. It's called all. I'll give you the main concept. What is all? Acute, you can write the full form. Acute lymphoblastic leukemia. Now what causes leukemia in human body? WBC. White blood cells and blood cells, they are defective. The person suffer with blood cancer. Right? Leukemia cases are there. Acute lymphoblastic, LYMPHOBLASTIC, lymphoblastic leukemia. See, acute and chronic, there are two words. Acute is short term, chronic is a long term. And lymphoblastic means lymph. WBC is formed in lymphocyte. Right? There it forms. Now, what is basically here? There is here, yes. So here genetic modification of T cells. What the scientists do there? They take out the blood, they take out the WBC, T lymphocytes, and they modify it in the lab. Now, genetic modification of T cells, there's something called this one, chimeric antigen receptor. See, cell has got, let me explain here. When you talk of cells now, I'll just take it upward. See, when you talk of cell has got something called receptors. On the surface of the cell, there's some kind of receptors are there, right? Like when you talk about advanced biology, there's something, advanced biology is something called cell signaling. Cells can signal each other. And that happens through some kind of a marker. What is a marker on the cell? So when you say chimeric antigen, is a protein. These are on the cell, what is called G protein and all. So what they do in the lab, they modify it. That's the name is CAR, C-A-R, CAR, chimeric antigen receptor. That is modified in the lab, right? But again, remember that here, this method does not use CRISPR-Cas9. See, what we have discussed, this one is a CRISPR-Cas9. Here, the direct use of CRISPR-Cas9 has been done. In this case, we don't use CRISPR-Cas9. Different way of editing, right? So here, genetic modification of T cells, chimeric antigen receptors to act as a kind of a, you can say to act as a cell that can kill tumors. Imagine that after modification, this T cell becomes like a kind of weapon. Are you able to understand? What they're modifying? T cell, which is a WBC, and this WBC will kill the tumor, kill the blood cell cancer. That means you are making a weapon to kill the tumors. That has been done through gene therapy, right? So please remember this one. This method, what I mentioned, is a very important thing which you have to remember that. Now, all are written here. Now let me ask, if you have written, I'll write a question here. And what I'm planning tomorrow, today after the class, I'm going to take the questions there. I'll collect some 20-25 questions and I'll give it to you tomorrow in the morning in the classroom. And we'll discuss through the question also the approach, how to solve such question there, based on the topics which are given there. I'll be referring to that one. So those who are online, those who are not in the classroom, they will get the soft copy. And those who are coming to the class, you will get the hard copy, printed form. And I will also discuss certain more areas. And what I would like to add here, because many things I have covered in science and technology in previous classes, but I will be dealing with the ecology, environmental aspect. And I'm planning to go through even the mapping part also. You have done some mapping classes here in Amigos, certain things? No. So I'll go into that. Ramsar site, some important species in the news, right? National parks, sanctuaries, biosphere reserve, Ramsar sites, which are in the news, we'll cover in the classroom there. So that, because you know, 2 questions, at least 2 or 3 questions are asked every year in the prelims exam about those kind of mapping part. Plus, you know, species part. We'll look into that. And what I'm planning that I can give you quick revision of the concept. That means the classroom you'll revise entire concept one by one. We'll be in fast mode. We'll do in a very fast mode, but you'll be able to revise everything there, right? So this part is okay there in biotechnology part of CRISPR-Cas9 and this part, which have made headlines, right? Now let's go to the another part of it. So this is where I have come across there. Now let me minimize it here. I'll go to that another document. There's a document here. Don't write, I'll be giving you this one. But first let me explain there. Then I'll give you writing part of it. What exactly you have to know there. Let me explain certain things. See here what you see, it is not very clear to everyone. I would like to just see there if I can select and will not be there. Okay, let me do one thing here. I'll write on the board also. One area which I think could be very important for the examination, right? That is the optical communication. I think last time I discussed the optical communication. Certain areas I have discussed there, but I'll bring into that. What you see the article here, let me first explain that article, then I'll give you the key points of that. See, when you talk about communication in day-to-day life, we use radio waves, right? Even in the space, you know, space communication. You know, NASA has got number of spacecraft, right? Even India, ISRO is using number of kind of a thing there, Chandrayaan and all mission. And we are able to communicate through the electromagnetic waves. So mostly what is used? Radio waves, long distance. Even, you know, mobile phone, we use radio wave. Now what is the emerging area? Light. Light as a mode of communication, medium of communication. So we can use in space, like outer space. We can also use in our day-to-day life. So what you find here, the first article, Deep Space Optical Communication by NASA. It has done for Psyche. There's a mission of NASA called Psyche mission. Anybody in the classroom, what is this mission aim for? Psyche mission. It is to study asteroid. Where do you find asteroid in solar system? In our solar system, where we have asteroid? Jupiter and Mars. So there is an asteroid made of iron and nickel. Iron and nickel. Now you know, have read in geography that our Earth is made of 3 layers: core, mantle, crust. And you know that crust is made of silicon and aluminum. Mantle is made of silicon and magnesium. And our core is made of Nephe. What is Nephe? Nickel and iron. Now, same way, Other planets also have got crust and mantle and core. See, every planet, what is planet? Is a hard body, right? It will have this kind of layer. Interior, what do you have? Core. Core of any planet. Jupiter, Mars, any kind of thing there. And what is a very unique thing about the core of the planet? Again, nickel and iron. Our Earth has got nickel and iron. Now, what is the mystery here? You have read in geography class, solar system, that asteroid would be remnant of some big planet earlier time, which got torn apart, broken by the gravitational force of Jupiter. Jupiter is a very, very big planet, which broken that part, rocky planet, what we call as a asteroid, and they go around the sun. So suppose, just do the assumption that asteroid which is today was a part of a planet. Just assume that earlier it was a planet. So what does that mean? If it was a planet, so what does that mean? It means that as part of the planet, it will have this nickel and iron. And let's say this got broken. This is broken into asteroids. So some part will be also part of the core. Suppose that. Imagine that this planet which was earlier between Jupiter and Mars was broken apart. So there will be part of crust, mantle and core of the planet. So then this core would have been also broken. So there is a asteroid called Psyche. Psyche. Now this asteroid is made of nickel and iron. That means which can give us understanding that earlier the asteroid was a part of a bigger planet. So there's a NASA mission Now I'll explain why it was in the news. One is this mission of NASA, Psyche mission, to study this asteroid which is made of nickel and iron. Right? Now, this spacecraft, how do we study the Psyche? NASA will send some kind of a spacecraft to study that particular asteroid. Now, you know that any spacecraft, whether it's a moon or Mars or Jupiter or asteroid or Saturn, has to communicate back to the Earth ground station, NASA center. And they require what? Communication system. So what NASA did, it used near infrared. Let me write here. NASA used near infrared light to communicate with spacecraft. That means what is something called deep space optical communication. Did you understand? Deep space optical communication. Not using radio wave, rather using light, infrared part of, you know that Vibgyor, violet, indigo, blue, green, and after red comes infrared. The near infrared light was used for communication. Now my question is that this type of communication, deep space optical communication, has what type of benefit? Now tell me, what type of benefit will be there? If we use light, near infrared, to travel along the space, vacuum, so what benefit will have? Instead of using normal radio waves, right? First time NASA used this light as a communication part. So what all benefits will be there? One is this one. Fast mode of data transfer. Communication with the spacecraft will be fast. You know, spacecraft need to be commanded from the ground station. You know that every space agency, whether it's a NASA or JAXA or ESA or ISRO, that need to command the spacecraft, it will be very, very fast. Second thing is what? Second thing is that distance. Travel in space. Through optical communication, space agency can transmit data to a long distance. You know that you have read in light years, the light which can travel very fast, and the information can be passed on. So this is what has been done. Now, see, this is a space mission. What I'm talking about, this is a space mission. But we can use the same type of technology on the earth in our communication part. So what is the future? That future we will be using light as a mode of communication. Today we are using as a radio wave, but in future we can use that. And you know that Li-Fi, there's a word called Li-Fi. Okay, I don't know whether in the last time I have discussed, first time in the country, Ladakh has developed a optical communication system. I think I would have covered there. Free space, right? Optical communication. So what is you have written this one? Shall I change this one? If you're done, then I'll change the page. All right, so I'm coming to the next one here. So what I would like to add here, this I have discussed already last time, free space optical communication. Now this is being also done in Andhra Pradesh, AP in Telangana, and also now in Ladakh they have done there. So light will be the part of the communication part. And generally what they use, laser light. Now this will be put just like a mobile tower. Like here you have a transmitter receiver. The dedicated transmitter receiver which will be able to send the light as a signal and receive the light as a signal. That way we can have open space communication. Even we can use satellite for the communication. That means it can be used on the building top, even aircrafts, and even the satellites can be used. We can use either on the ground, or we can use on the top of the building tower, right? Or we can use in a moving aircraft. They can use this kind of system, and even we can use in satellite. So this technology will be scale up. This technology is going to scale up to a larger level there. And now what I've given the example of the NASA deep space. They're using for deep space. Correct? So that part has come. So now let me get back to here. What I've explained here, this part. So this is what is given here, optical communication part. What you find here, I have explained that. What is the mission? There's Psyche mission. Now, okay, let me explain here. Then you can read from that will not write tantalum. Or let me write here, tantalum. What was in the news here? There's a metal called tantalum. Anybody where you have read about tantalum? It was in the Hindu paper, Indian Express. See, one thing which has made a headline about lithium, lithium deposit in Himalayas, right? Ladakh and this region. Another thing which has made a headline about tantalum. Okay, let me give you the main points of that. IIT Roorkee, right? IIT Roorkee, a team of scientists from IIT Roorkee, they have discovered tantalum in the sands of a very important river in Punjab, Satluj. Indus has got 5 tributaries. Satluj is one of the tributaries of Indus. So here found tantalum in sands of Satluj River. This discovery has been made, right? This discovery has been made about this tantalum here. that this tantalum we have a deposit. It was a new discovery made there, and tantalum now is, remember that it's a rare, with something called rare earth metals. Now let me ask the chemistry behind it. Where do you find rare earth elements or rare earth metals? Where in chemistry, if you look at periodic table, so where you come across such kind of metals? There's something called here F series of the modern periodic table. There are two things are there. One is called lanthanide, and another series you'll find actinide. Now when you talk about tantalum, that is a part of lanthanide series. Lanthanum has the atomic number. How the periodic table is arranged in terms of elements with increasing atomic number. So lanthanum has got 57. Based on that is been arranged there. So it's basically here part of lanthanide series. See, earlier it was considered to be niobium. There is a metal called niobium. But later on scientists discovered that it's not niobium but a new metal is there, which was given a name after a Greek king called Tantalum, who was cursed to go under the earth and stand in the water. From there the name Tantalum has come, right? So here part of lanthanide series. So where the use of lanthanide series there? Always remember that whenever you come across lanthanide series, the first application electronics, semiconductor, semiconductor electronics. There this particular lanthanum based, this tantalum can be used in the electronic industry. So that discovery can really help India in terms of extraction of this metal and use in electronics. Now another thing is that is a hard metal in oxide, when we form oxide and carbide form mixed with graphite. If we mix with graphite, I'll explain here what is the thing here. If we make tantalum oxide or tantalum carbide and react with graphite, it becomes hardest metal. You understand? Hardest, which will not be broken. When it becomes hardest, tantalum oxide with graphite or tantalum carbide with graphite, it becomes so resistant that nothing can happen to this metal. It's such a metal is there. Now another very important thing is a ductile. What is the meaning of ductile? Is a ductile property. What is ductile property means? When you read in chemistry, which can be drawn into, which can be molded into wire. Copper wire you have there. So even it has a property of ductility. So UPSC will not ask you the discovery made in Satluj river. There's a part of geographical aspect. You should know what type of metal where it can find the application part. So that was one of the things which was in the news, because I have found mostly the current affairs have focused on lithium ion. And now again, lithium ion is also very. Now let me ask there. Is lithium ion rare metal? Lithium ion. Today we use the battery, no? Our battery, laptop, mobile phone, all these lithium ion batteries, the rechargeable battery. Is it a rare earth metal? No. Lithium is a normal metal. Sodium, potassium, calcium, there comes, and lithium comes in which group? Which group? Sodium, sodium, potassium, you have all the names, no? Calcium, so which group it comes? Lithium. Correct. Lithium, sodium, cesium, that same series comes there. Cesium is the last. At the bottom you will find. Now this cesium part, if you look at, after that there is an actinide. This is called actinium. These are radioactive. Uranium, plutonium, thorium. This all comes in actinide series. But yes, lithium is a normal metal, just like sodium, potassium is a normal metal, which we find in the battery system there, right? So this is what has come into the news there. So is it clear, everyone? Right? I'll come to the next article. Any doubt you have? You're able to follow? So if in case surprising question comes in the exam about tantalum, you should know these all points there. Now I have written F series. In chemistry, we've written 4 are there. S, P, D, F. S, P. S is mostly metallic metals. P is you'll find non-metals. and D is called something called transition metal. And when you say F, lanthanide, actinide, they're called inner transition. In chemistry, we'll read about inner transition metals. So they belong to there. Tantalum.